Okay, hi everyone. This is Lecture 13, Part 1 of the Endocrine System. So today we are going to introduce you to the basics behind how the endocrine system regulates homeostasis. We have just finished the nervous system unit, and to compare the nervous system to the endocrine system, the biggest difference is really the time scale. So the nervous system acts very quickly through electrical signals. The endocrine system acts very slowly on a long time scale through hormones. When we say long time scale, we mean hours, days, sometimes even weeks to make lasting changes in the endocrine system. So hormone is a chemical. Specifically, it's a chemical signal that is released into the blood to regulate distant target cells. So there will be an endocrine gland that releases a hormone, a chemical, into the blood. It's going to travel through the blood, through the entire bloodstream, and it will then reach its target organs and target cells. So endocrinology is the study of hormones and the endocrine organs that produce and regulate the release of hormones. So overall, there is a lot that the endocrine system can regulate, and it all comes back to the basic homeostatic systems in the body. So it can regulate reproduction, can regulate growth and development, can regulate electrolyte, water, and nutrient balance in the blood. It can regulate metabolism and energy balance, and it can also regulate the immune system. And sometimes the endocrine processes will regulate multiple of these examples all at the same time. So because growth and development requires a lot of nutrients, sometimes you will not only regulate growth, but you will also regulate metabolism. So these all can be linked as well. So to review from anatomy, there are major endocrine glands starting in the central nervous system. So the hypothalamus, the pituitary gland, those are these small nervous system regions down here, lower down in the lower regions of the brain. In the back of the thalamus, there is a tiny gland called the pineal gland that we will also talk about. And then we get to the peripheral endocrine organs. We have the thyroid gland located here in the neck region, and the tiny parathyroid glands that are on the back of the thyroid. We have the thymus gland that is not visible in adults but will be visible in babies and small children. The adrenal glands, which sits on top of the kidneys. The pancreas, which has other functions, namely digestive functions, but it does have major endocrine function. We also have the reproductive organs. Primarily the gonads are responsible for the reproductive hormones, and that is in females the ovaries and in males the testes. So endocrine glands are different from exocrine glands. So we learned about, for example, sweat glands and oil glands, your sebaceous and sudoriferous glands that are located in the skin. Those glands also secrete chemicals, but they will secrete chemicals to outer surfaces or to outer body regions. The endocrine glands are secreting inside. So endo means inside. So they're secreting into the body, and those secretions get into the blood. They're going to travel through the blood to regulate distant target cells. And the target cells will respond to the hormones only if they have receptors for that hormone. So I'm going to draw this out so we can start to build on this bigger picture for you guys. Okay, so what we have then is we're going to have a gland. We're just going to loosely call it an endocrine gland right now. We will give them specific names as we go through. We're going to have an endocrine gland that contains secreting cells, and those cells are going to secrete into the space outside of that gland. that is not connected to the outside of the body. It's going to go into the the extracellular fluid in between the cells. And then nearby, that hormone will be picked up by the blood. So it's going to enter into the blood, these various chemical hormones, and they will travel through the blood, reaching various targets. We're going to talk today about how target cells respond to hormones and how non-target cells can avoid responding to hormones. But mainly the idea here is that you'll have target cells that will have receptors for that specific hormone. And as the hormone travels through the blood, it will reach the receptors on the target cell and make changes to the target cell. Nearby, there may be other cells that are non-targets or that we do not want to be regulated by those hormones. Those non-target cells will have different receptors that do not recognize the hormone. So even though the hormone may be nearby, it will not bind to those target cells. And in the non-target cells, we will have no changes. So hormones are specific. They're produced by endocrine glands. They enter into the bloodstream, and they affect their targets through specific receptors. The mechanism of action then for a hormone is through these hormone receptors. So a hormone receptor, just like any receptor on a cell, is a protein that recognizes and binds to a specific chemical, in this case a hormone. Hormone receptors, just like other receptors we've learned about, can either be extracellular, meaning they're located on the plasma membrane, and they're linked to the inside of the cell by nearby molecules through second messenger systems. Or they can be intracellular receptors, and they're located inside the cell, and usually they will then directly activate genes. These intracellular receptors will be at or near the nucleus and near the DNA to directly change gene activity. The target cells then have to have receptors for that specific hormone so that they can change cellular processes when they get the hormone signal. If they do not have the receptor, they will not change in response to the hormone nearby. So let's remind you of what types of hormones can get into the cell and what types of hormones cannot get into the cell. So amino acid based or water soluble hormones, can they get into the membrane or not? Remember back to our membrane transport lecture. No. So amino acid based and water soluble hormones are polar and large, meaning that they often have charged regions and or they are very large proteins. They can range in size from large proteins to polypeptides. They can also be specific amino acids. In any case, they are water soluble and they cannot cross the plasma membrane. So in other words, they are hydrophilic and they will stay on the outside of the membrane. So they'll travel through the bloodstream and they will be stuck outside of the cell. They cannot get into the cell. So they have to act then through extracellular receptors. So these extracellular receptors will be on a specific target cell, and then they will be linked to G proteins and a cascade of other molecules within the cell that will actually cause the changes. Steroid hormones and related hormones are lipid soluble. Steroid means that they are derived from cholesterol, and if they are derived from cholesterol, then they are lipid soluble hydrophobic. If something is hydrophobic, it can get through that region of the membrane, not drawn here, but that region of the membrane that has the hydrophobic tails. So they pass through the membrane no problem, and they will interact with receptors inside the cell. It doesn't mean you can't have extracellular receptors for a steroid hormone, but it just means they don't need it. So the intracellular receptors then are much more direct. They're going to be at or near the DNA. And once the intracellular receptor binds to its hormone, it can then affect protein synthesis and DNA activity directly. So if none of this sounds familiar, you may want to go back to the membrane transport lecture and review membrane selectivity and phospholipids, where we see that lipids can cross easily across the phospholipid bilayer, but polar or large molecules, water-soluble molecules, have to have proteins that they interact with. I also want to remind you guys of the hormone mechanisms of action. We did this in the cell communication lecture, but that was lecture number 7, so it's been a few lectures since then. So just a brief review of the hormone mechanisms of action. We can either have second messenger systems or direct gene activation. So a second messenger action is when a water-soluble or amino acid-based hormone cannot get directly inside the cell, meaning they rely on these extracellular receptors. And then the change in the extracellular receptor that happens from the binding of the hormone will change a domino effect of molecules within the cell. The first thing that changes will be the G protein coupled receptor, and then several other downstream molecules. The last and final molecule that it affects before it makes major changes to the cell is called the second messenger. And we have 2 second messenger pathways, the cyclic AMP pathway and the calcium-calmodulin pathway. So in the cyclic AMP pathway, the hormone will bind to a receptor. It's a G protein coupled receptor, meaning that when the hormone binds to the receptor, it makes a change in that receptor. We call that a conformational change from the binding. You get a shape change in the receptor. When it does that, that bumps the G protein and activates the G protein nearby. That G protein then is activated and will activate another nearby molecule called adenylate cyclase. Adenylate cyclase then will make the second messenger cyclic AMP, and it makes a lot of it. This is what we call signal amplification. So one single binding of a hormone to a receptor makes a lot of this second messenger. So it doesn't take a lot of hormone binding to get big changes in cyclic AMP. Cyclic AMP will then activate a series of molecules called protein kinases. Protein kinases are going to add phosphate bonds, we call that phosphorylating proteins, inside the cell and can then change the activity of various proteins inside the cell. So for example, you could have some enzymes. You phosphorylate those enzymes, you get more activity of those enzymes. You could have some receptors. You phosphorylate those receptors, suddenly you insert more receptors into the membrane. Channels, you phosphorylate a channel, you get channel insertion into the membrane, and then you can get large changes inside the cell. These are very general pathways. So I always tell my students, we would go to these biochemistry lectures when I was in graduate school, and it's sort of like blah, 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 activates protein kinases, makes many changes inside the cell, blah, blah, blah. So if you ever want to sound smart with a biochemist, then you say, oh yeah, cyclic AMP activates protein kinases. Yep, makes a lot of changes inside the cell. And that's so common that pretty much will be like, oh, okay, yeah, that does, that works. A lot of changes inside the cell. So protein kinases through these second messenger pathways are ultimately what cause the changes inside the cell. And it can be any number of things. It can be as diverse as the protein classes are within a cell. The calcium-calmodulin pathway is very similar, but the names of the molecules are different. So same idea, hormone binds to the receptor activates a G protein, the G protein then instead of adenylate cyclase will activate something called phospholipase C. That makes something called PIP2 and diacylglycerol or DAG. It will also make IP3, inositol triphosphate. Don't be intimidated by these words. PIP2, DAG, and IP3 are just downstream molecules that activate protein kinases and make changes inside the cell. So DAG activates protein kinases. IP3 activates protein kinases, but before it does that, it goes through calcium first. So IP3 causes calcium release. Calcium binds to another molecule called calmodulin and then activates protein kinases, makes a lot of changes inside the cell. Okay. So don't be intimidated by these cascades. There are several molecules, but you can draw a simple diagram for yourself, linking just to memorize quickly each of these molecules. Here is the diagram that your book provides for the cyclic AMP pathway. Actually, I think this is from a different text. Yeah, this is from a different textbook, but I chose it because I like it better than what's in your book. But you have something very similar in your book. So the hormone, here's the hormone in pink here, binds to its specific receptor, activates the G protein. The G protein is inside the cell now and can make changes to other molecules inside the cell. The G protein will activate adenylate cyclase. Adenylate cyclase will then make cyclic AMP, a lot of it. So really this diagram shouldn't have just one cyclic AMP, it should have many cyclic AMPs, but for simplicity they've just drawn one. Cyclic AMP then will activate protein kinases, which will make a lot of changes within the cell. Okay. In addition to water-soluble and amino acid-based hormones, the other class of hormones will be the steroid-based or lipid-soluble hormones. These have an easy route into the cell. They can just get straight through the membrane and activate an intracellular receptor. So this is quite simple. The receptor is in the cell. The receptor is in the cell, binds to the hormone, and will change something in the DNA. So here's the steps for direct gene action. The steroid hormone diffuses into the cell right through the plasma membrane. The receptor inside the cell forms a receptor hormone complex, so they stay together. The receptor hormone complex then will either be very nearby or will move to the DNA to bind to the DNA. Once it binds to a particular region of the DNA, it will be specific to a particular gene or a particular set of genes. It will activate transcription. What is transcription? Transcription is converting DNA to mRNA. You guys should remember this from our DNA lectures. Once we have mRNA, mRNA will leave the cell and leave the nucleus and be translated into proteins within the cell. So rather than having protein kinases that are phosphorylating proteins already made in the cell, which is what happens with second messenger systems, direct gene action can actually make new proteins. Protein changes activity within the cell then will depend on the specific genes or regions of DNA that are activated by the receptor hormone complex. So for example, we could increase secretions. We could increase more signaling molecules. We could increase receptors. We could make more structural proteins. We could make more secretory proteins, any number of things. Again, as many different types of protein classes that are within the cell could be the range of activity that's changed by direct gene action. So ultimately, both the water-soluble and the lipid-soluble hormones are changing proteins within the cell. But the water-soluble hormones have to change proteins that are already present. The lipid-soluble hormones can actually lead to direct synthesis of new proteins. So now that we have the hormone receptor interaction, let's talk about how we can modify the target cell response based on these hormone receptor interactions. So both hormone and receptor levels can be changed. So the level of hormone that's in the blood can increase hormone activity. The level of receptors that are on the target cell can increase hormone activity. And how strongly the hormone receptors bind to each other, or the affinity of this interaction, the more strongly the hormone binds, the longer it will be bound, and the more likely it will be to bind, the stronger the hormone action. So first, let's talk about upregulation versus downregulation of receptors. Usually this happens when you have chronic levels of a hormone, and just like the nervous system ignores things that are present all the time, signals that are present all the time, the endocrine system will begin to respond less to hormone signals that are there all the time. So that would be downregulation. So we have chronically high levels of a hormone, and the cell will then start to decrease receptors for that hormone so it doesn't respond as much. So basically, this is the cell getting bored, right? Yeah, we've heard that protein signal a million times. I'm going to take out my headphones, right? So that's downregulation. We can also have upregulation, where we can increase receptors for a specific hormone in order to increase the response to a hormone. So let's draw this out so you guys can picture what this would look like. Okay, so again, here's our previous drawing, right? You've got the hormone released, the hormone in the blood, and it's going to go to target cells. Now, some of these target cells will have a different level of receptors. So if I now draw a bunch of receptors on this cell compared to this cell, which one is going to respond more? Right, the one that is upregulated. So upregulation means that we add more receptors, and now this cell will be able to receive the signal more from that hormone that is in the bloodstream. If we take this situation though and we remove receptors and go back down or downregulate, we decrease the number of receptors for a specific hormone in a target cell. This can happen through endocytosis, degradation of receptors, decreasing transcription and translation, etc. So we remove receptors, then the hormone might be nearby, but it's reaching its sort of maximum activation because it's only got one receptor to activate. Once that receptor is bound, the other hormones are not going to have anything to bind to affect this particular target cell. So upregulation, many receptors, big response. Downregulation, fewer receptors, decreased response. So we can also control the release of hormones. The amount of hormone that is being synthesized and released is tightly regulated for homeostasis. Negative feedback here is very common, and the basic idea is that you have a stimulus to an endocrine gland. When you stimulate that endocrine gland, you produce more hormone. There are 3 ways to do that. You can track the levels of ions and nutrients in the blood, and the direct level of ions and nutrients in the blood will stimulate the gland. So for example, if blood sugar is too high, you might have certain hormones released from the pancreas. If blood sugar is too low, you will have other hormones released from the pancreas. So the stimulus to the gland in this case is through direct levels of molecules in the blood. This is also called humoral stimuli, but be careful not to mix that up with hormonal stimuli. So the blood or humoral stimuli is the level of certain molecules in the blood that is being sensed by the gland. And that gland then will produce hormones based on the levels that it senses. There's direct electrical stimulation of certain glands. That's neural stimulation. So electrical stimulation to a gland can, through nerve fibers and electrical stimuli, increase the production or release of a hormone. Finally, some hormones regulate other hormones. So one hormone can cause the gland to be activated, and that gland will then produce a second hormone. In either of these three cases, through the blood, through the electrical or neural, or through other hormones, the hormone production will increase, and that level of hormone will send a signal back to the gland to say, okay, we have enough, you can slow down now. This negative feedback then will keep the levels of hormone balanced. So here are some pictures and examples from your textbook. No, actually, this is from a different text. I wasn't crazy about how your textbook did endocrine system, guys, so I chose these from a different one. So hormone release can be caused by see, I did it too. Hormone release can be caused by blood or humoral stimuli. In other words, here's one. Calcium affects the parathyroid glands. When calcium is low, the parathyroid glands will release parathyroid hormone to bring calcium back up. Then the level of parathyroid hormone will come back and signal and say, yep, we've got enough parathyroid hormone, you can now slow down your release. Neural stimuli is when you have direct neural activation. So here's a nerve coming down from the autonomic nervous system to the adrenal medulla and stimulating electrically release of epinephrine from the adrenal medulla. Then here's hormonal, where you have hormones from a higher up gland dictating and increasing or decreasing the release of hormones from other glands. This is a more complicated situation, and we're going to talk about this toward the end of this lecture. So blood stimuli is levels of ions and nutrients that can trigger a gland to produce and release more hormone. Here's the example. Low blood calcium triggers the parathyroid gland to release parathyroid hormone. Parathyroid hormone then will balance that low blood calcium by increasing blood calcium, and the levels of blood calcium will then come back up, and as those levels come back up, it'll turn off the parathyroid gland signal. Neural stimuli is direct neural activation, electrical signals to the adrenal medulla, for example. Adrenal medulla then will release epinephrine and norepinephrine. In this case, the end of the neural stimuli is the end of the electrical, whatever caused that electrical signal. In this case, the electrical signal came from the sympathetic nervous system. So this will end when the stressed state of the sympathetic nervous system is over. For hormonal stimuli, we have several levels of glands that can cause signals to glands lower down in the chain. So we'll start with hypothalamus, which makes something called TRH. That hormone stimulates the anterior pituitary to produce TSH. TSH then, a second hormone, will cause the thyroid gland to produce the final hormone, thyroid hormone. So this is actually two levels of hormonal stimuli, hormone to the final release of the final hormone. And then the levels of that final hormone will signal back to the upper glands that they can slow down their activation. So the complexity of this hormone to hormone release gets a little bit easier if we talk about what these names of hormones mean. So there's something called tropic hormones. When you hear corticotropic, gonadotropic, when you hear the word tropic within the name of a hormone, what that hormone does is it stimulates the production and growth of another gland. So if something is tropic, it increases growth. In other words, it's going to send signals to another gland to grow more and produce more. And because that other gland is an endocrine gland, it's going to grow more endocrine product. It's going to increase the activity of those cells and produce more hormone. You will also see releasing and inhibiting hormones. Releasing and inhibiting hormones don't affect the growth or the cellular activity. In other words, they don't affect the levels of the hormone being produced, but they affect the release of the hormone. So we can grow and produce a ton of hormone, but it's still stuck there. We then need to give a signal to tell it to let go and send it into the bloodstream. So releasing hormones will stimulate the release of a hormone by another endocrine gland. Inhibiting hormones will stop the release of a hormone by another endocrine gland. So this, in my mind, is like your manager, and then your assistant manager, and then the final endocrine gland is the worker. So the manager sends out the, hey, you guys need to get to work and start producing. The assistant manager says, yep, you've produced, now go, do what you were supposed to do. And then the gland does the work. The worker does the work. So manager, tropic hormone, assistant manager, releasing hormone, actual worker, the person who does the work, is the endocrine gland. We also have changes in receptor level responsiveness. So when hormones regulate other hormones, they can change the responsiveness of receptors. So this is kind of like the releasing inhibiting idea. But there are two ways to affect the release. So one way can be permissive. So this is saying that the presence of one hormone allows the second hormone to respond. So this is like the manager saying, okay, you can do your job. And by doing that, it's going to increase receptors at the target cell. Then there can also be synergistic effects, which maybe is two co-workers acting together to get a job done. So in this case, we'll have two hormones combined to increase each other's action. So they're helping each other. And they would get more done than if they worked by themselves. So this is when the two hormones from two different endocrine glands, or maybe two hormones from the same gland but different overall structure, will combine together in a cell or in a target cell to get more interaction of the final effect. We can also have antagonistic responses. That is that one hormone stops the other hormone from working. And that is to reduce the effectiveness of another hormone by decreasing receptors for that hormone. So permissive and synergistic type hormones can upregulate receptors at the target cell. Antagonistic hormones will downregulate receptors at the target cell. We can also change how a hormone is concentrated and how it is actually released. So the concentration of a hormone in the blood depends on whether it is lipid or water soluble. So let's go back to this diagram. Let's turn this on. So we talked about the stimulation of the endocrine gland by neural, hormonal, and blood stimuli. That will then affect the release of the hormone into the bloodstream. So maybe we should write that up here, right? So there is a stimulus, neural, hormonal, or blood stimulation of the endocrine gland. Once that release happens, though, we can also affect the concentration of a hormone. So there are proteins in the blood and there is plasma in the blood. So if a hormone is lipid soluble, meaning it's hydrophobic, it's not going to be dissolved in the plasma. So we have plasma, which is mostly water. And then we have proteins floating within the plasma. So a lipid-soluble hormone cannot dissolve in the plasma. It's going to bind to proteins in the blood. A water-soluble hormone we consider to be freely circulating because it can just float around and be dissolved in the plasma. So lipid soluble hormones have to be bound to proteins in the blood. Water soluble can dissolve freely in the plasma. We can also affect the timing of a hormone by how fast it is released to the stimuli and by how fast it is degraded by enzymes on the back end. So as the hormones travel through the bloodstream, they will reach the liver or the kidneys, which can break them down and reduce the overall concentration of the hormones in the blood. Okay, so hopefully you guys are getting a picture of everything that has to happen for a hormone to reach its target cell. There's a stimulation that can happen through various different stimuli to the endocrine gland that is producing that hormone. Once the endocrine gland gets the appropriate signals and produces that hormone, then there are signals to release that hormone. That hormone will get released into the bloodstream and can travel either bound to plasma proteins or dissolved in the plasma. Then it's going to travel through the bloodstream and look for targets. The target cell response then will change based on the receptors that are available in the target cell. More receptors, more affinity for those receptors, bigger response. Fewer receptors, less affinity for those receptors, will be a smaller response to that hormone. And we can regulate at all of these different levels to ultimately affect the endocrine response. So now we're going to get into what is actually happening in each of these glands. We're going to start at the hypothalamus and work our way through. We're only going to get through a couple of them in this lecture, and then we will complete the rest in part 2. So we're going to start at the brain region that really controls and regulates most of the other endocrine organs. So this is the hypothalamus. So we always say that the hypothalamus is our homeostasis region. It's doing a lot neurally to regulate homeostasis through the nervous system, but it's also doing a lot hormonally to regulate hormone function. So we call this a neuroendocrine organ. But it's within the brain. It is a brain region that has endocrine function. So it's a neuroendocrine, I guess organ is okay, right? For lack of a better word. The hypothalamus produces 9 hormones. And I want you guys to write each one of these down fully as we go through their names and little by little start a chart to memorize what each of these hormones does. We're going to start with the first 2, ADH and oxytocin, and then we'll go through the rest, which are our releasing and inhibiting hormones. The pituitary gland is attached to the hypothalamus. So here's the hypothalamus. If we want to go back to overall where it is in the brain, do I have a picture of the brain? Yeah, I don't have a picture of the brain. Okay. Where it is overall in the brain is in this lower region we call the nose of the seahorse. So you guys can go back to your brain anatomy to find the hypothalamus just below the thalamus. So here, highlighted in green, is the hypothalamus, and then hanging down from the hypothalamus is the pituitary. The nose would be in this direction, so the nose of the person, so the front or the anterior portion. So your patient is facing this way, I think. So for this, we have a front and a back to the pituitary. We have the anterior pituitary and the posterior pituitary. And they really are two very separate and distinct endocrine organs. The anterior pituitary is glandular, and it's also called the adenohypophysis. It produces and secretes 6 different hormones. The posterior pituitary is actually mostly neural tissue, and it doesn't produce any hormones, but it can store and release hormones. So first, the hypothalamus to anterior pituitary. The hypothalamus is connected to the anterior pituitary through a blood connection. We call this blood connection the hypothalamic hypophyseal portal system. So this is that you have, here's your hypothalamus, you have neurons in the hypothalamus that are producing hormones, and those hormones, hopefully you can see these sort of green and red and blue dots that are then passing through the bloodstream, through this hypophyseal portal system, and going down to the anterior pituitary. So we have hormones produced in the hypothalamus passing through the blood, so these are true hormones going straight through the blood, and then making changes in the anterior pituitary. So their targets are in the anterior pituitary. The hypothalamus to posterior pituitary is a neural connection. And really, the posterior pituitary is simply an extension of the hypothalamus. So we could say, but the anatomist didn't, but in my mind, it's almost like the hypothalamus stretches down like this and has its own separate little arm, which is the posterior pituitary. So you have other neurons in the hypothalamus that are making hormones. Here are these green and red dots for different hormones, and their axons are going to carry those hormones to their axon terminals, where the hormones will be released into the blood, leaving the posterior pituitary. So the posterior pituitary is going to store these hormones until it gets signals from above to release them into the blood. So really all you have in the posterior pituitary are axon terminals and hormones stored that was produced in the hypothalamus. So here's a closer view of the hypothalamus to the anterior pituitary with the hypophyseal portal system. And then here, if you look at the histology, you can also see a big difference. You guys probably did this in anatomy, but the anterior pituitary looks very glandular, and you can see the density of cells there in this dark purple staining. The posterior pituitary does not look very glandular, and in fact, it's usually very light staining because it's really only axon terminals, not a lot of cell bodies here in the posterior pituitary. So what is actually being made in the hypothalamus and sent to the anterior pituitary? The hypothalamus is going to secrete hormones that regulate the anterior pituitary. So the hypothalamus is secreting, releasing, and inhibiting hormones. So these are hormone to hormone stimuli. So when you see releasing hormone or RH, so for example, you will see thyroid releasing hormone or thyrotropin releasing hormone. That's an RH. What that means is that is the hypothalamus telling the anterior pituitary to release a hormone. So this is the manager telling the assistant manager to send out his commands. So releasing hormones increase the release or secretion of other anterior pituitary hormones. Inhibiting hormones are the manager telling the assistant manager, nope, don't send instructions yet. That's going to decrease the release or secretion of other hormones. The hypothalamus to posterior pituitary is a simple production and storage relationship. So let's do the hypothalamus to posterior pituitary because that's the easiest one. So the posterior pituitary does not produce any hormones. I'll say it one more time because students always get the anterior posterior mixed up. Posterior pituitary does not produce any hormones. It only stores and releases two hormones that are produced in the hypothalamus, and these two hormones are very interesting. The first hormone is called ADH, or antidiuretic hormone. It also is called vasopressin. You will hear the two names sort of depending on whether the researcher is more interested in its kidney function or in its brain function or blood vessel function. ADH and vasopressin are one hormone produced in the hypothalamus that travels through the axons to the axon terminals and gets stored in the posterior pituitary. The other hormone is oxytocin, produced in the hypothalamus, travels through the axons, and stored in the posterior pituitary. So let's talk about what each one of these does. So ADH is produced in the hypothalamus and released by the posterior pituitary. Its main targets, at least that we know of now, there's a lot more research going on with ADH, but its main targets and major changes in the body are actually the kidneys. So ADH, its function is to conserve water by preventing water release in the urine. When we get to the function of the kidneys and the specific structure of the kidneys and channels that are in the kidneys, we'll talk about how it does that. For now, I want you guys to know that it conserves water by keeping water in the body and preventing water release in the urine. So it's an antidiuretic. It stops diuresis or stops peeing. It will also have effects on the blood vessels. It can contract smooth muscle in the arterioles, which we call vasoconstriction, to overall increase blood pressure in the whole body. The regulation of ADH is through osmoreceptors in the hypothalamus. So osmoreceptors are sensing the level of water and solutes in the blood. So what kind of a stimulation is that? We have three possibilities, right? It's either blood stimulation, neural stimulation, or hormonal stimulation. So what kind of stimulation is stimulating ADH if it's osmoreceptors? Blood. Osmoreceptors are looking at levels of molecules in the blood, so this is a blood stimulation. So levels of molecules in the blood or level of osmolarity in the blood triggers ADH release. Also, blood volume receptors in the heart or the atria of the heart can trigger release of ADH, and namely if blood volume or blood pressure is too low. So vasopressin or ADH will be released. It can then conserve water or constrict blood vessels. The net effect of that will be to increase blood volume and increase blood pressure. The next hormone is oxytocin. Oxytocin is made by the neurons in the hypothalamus and released and stored in the posterior pituitary. Its targets are the uterus, the mammary glands, and we are now learning a lot more about the brain targets of oxytocin. Its function is to stimulate uterine contraction during childbirth, so uterine contraction during labor and delivery. So that pushes the baby out as the baby is pushing. This was actually the positive feedback loop that we did way back when. It can also promote milk release during breastfeeding, so lactation. And it's now being studied in its role in social behavior, particularly maternal behavior, maternal bonding, and attachment. As an aside, oxytocin is also often called the love hormone because it's released during sexual activity, particularly during orgasm. So oxytocin sometimes is the love or orgasm hormone, which relates to its role in social bonding and attachment. The regulation of oxytocin we know most about during childbirth, positive feedback regulation. And it is regulated by birth canal reflexes, or the stretch of the uterus during contraction. It's also regulated by stimulation of the mammary glands for milk release. So these are all hormones that are produced. ADH and oxytocin are hormones that are produced in the hypothalamus and stored in the posterior pituitary. Totally unrelated, right? Doesn't it seem like they're totally unrelated? Oxytocin, the love hormone, childbirth, lactation, social bonding. Vasopressin, blood volume regulation, kidneys, right? But the research actually on vasopressin is also showing social bonding and other roles for vasopressin and ADH as well. That's probably more to come, but keep an eye out for it. I'm sure you guys will see it at some point in your clinical careers. So the next is to start talking about what these specific hormones are and what they do. So first, from the hypothalamus to anterior pituitary, the hypothalamus releases 7 different hormones. Each of these 7 hormones goes through that hypophyseal portal system, or through that tiny bit of blood vessels from the hypothalamus to the pituitary. And these hormones are then going to regulate specific hormone pathways that are regulated by the anterior pituitary. So I think, again, we can use the manager-assistant manager idea. So these are management hormones, and these management hormones are releasing and inhibiting hormones. So you will see RH and IH in the names of these hormones. So TRH is thyrotropin releasing hormone. CRH is corticotropin releasing hormone, etc., all the way down the list. So these 7 hormones from the hypothalamus are going to travel through the infundibulum, through that blood supply, down that stalk connecting the hypothalamus to the anterior pituitary, to the anterior pituitary. And they will individually regulate these hormones that are produced by the anterior pituitary. So again, hypothalamus hormones, the managers, anterior pituitary, assistant managers. And the assistant managers are then going to be the hormones that directly affect the workers, or the endocrine glands. So the anterior pituitary then has 6 hormones that will be affected by the hypothalamus. Growth hormone, which can be affected by GHRH and GHIH in the hypothalamus. Thyroid stimulating hormone, which will be affected by TRH in the hypothalamus. ACTH, adrenocorticotropic hormone, which will be affected by CRH from the hypothalamus. Follicle stimulating hormone, which will be affected by the gonadotropin releasing, or GnRH, as well as luteinizing hormone will also be affected by GnRH. And then prolactin, which will be affected by the prolactin releasing and inhibiting hormones. I know, it's starting to get complicated, right? So what I want you guys to do is print out the chart that I'm going to link to this lecture. And in part 2, we're going to go one by one through the target glands, through the endocrine glands. What is it that the workers that are doing the work for homeostasis, what is it that they are actually doing, and how is it that they are being managed by the hypothalamus and anterior pituitary, and what are those endocrine gland targets? So get out a lot of notes, print out that chart that I'm going to link to this, and we're going to start little by little going through the individual endocrine glands, how they are regulated upstream by the hypothalamus and anterior pituitary, and then what they cause downstream in the target cells. So don't get freaked out yet, okay? This is a quick summary of what the anterior pituitary hormones does, but these will make a lot more sense after we give you the full pathway, okay? Come back to this after you do part 2, and this will help you just organize your notes. So the last thing is to look at the control of the hypothalamus. So the hypothalamus itself is also controlled. It has a company in charge of it, and that company is the brain. So there are diverse regions of the brain that can regulate the hypothalamus. There are emotional regions of the brain, the limbic system, that can affect hormone control. There are levels of homeostatic factors in the blood that can affect the hypothalamic control. And there's a lot of negative feedback from downstream hormones that can affect the hypothalamus. So the manager as well can be regulated. Okay? All right, I know this was a very dense lecture. This is the beast that is the endocrine system. Start little by little writing out the names of these hormones and print out that chart, and we're going to work on that chart in part 2. Let me know if you have any questions. I'll see you guys in a bit.